am going to talk to you all about the mechanisms of polyploidy today. To get started, I am going to tell you just some of the basics about polyploidy. A polyploid organism is an organism that has 3 or more sets of chromosomes, and because the organism has more than the diploid number of chromosomes, this causes the organism to have a change in genotype and phenotype. And the modifications generate morphological and physiological changes, which ends up changing the habitat and geographical dispersal of the organism, and also the reproductive and breeding systems of the organism. Polyploidy was discovered about a century ago and has been found to occur often in evolution. And they found that it is an ongoing process more than instead of a macroevolutionary event. And polyploidy is very important in organisms such as plants, fish, yeast, insects, reptiles, amphibians, and many other organisms. They found a single polyploidization event that happened about 1.5 million years ago, and this shows that polyploidy is very important because we still see it today. And they found that many plants are different from their parents in a number of ways, and this is contributed to heterosis, which heterosis is when hybrids tend to have a better overall performance than their parents. And the overdominance model shows allelic interactions within each of the many genetic loci that cause the better performance in the offspring. And they've also found that altered regulatory interactions play a role in heterosis. Polyploidy is very important to organisms that live in harsh environments, and it is also very beneficial to these organisms because of the phenotype change that the polyploidy causes in the organism. And an example of this would be like a cactus in the desert, which I will explain more later. And also, polyploidy leads to increased cell size and organ size, and it is an irreversible process which causes the organism to have an increased level of polyploidy throughout its life. There are 2 types of polyploidy. One type is allopolyploidy, and the second is autopolyploidy. Allopolyploidy is allopolyploids contain chromosomes derived from 2 different species, which is a result of the multiplying of chromosomes in number in an F1 hybrid. And there are 2 mechanisms by which an allopolyploidy can be formed. And the first mechanism is a 2-step mechanism, and this is when there is hybridization of 2 diploid species, and which is followed then by chromosome doubling. The second way in which an allopolyploid can be formed is through a 1-step mechanism, and this is when there's fusion of unreduced and male and female gametes. And they have found that the 1-step mechanism is more prominent in plants because they produce some unreduced gametes which can then fuse together to form the allopolyploid. And also the chromosome doubling that occurs in the 2-step mechanism is a rare event, so it does not show up as often. And this diagram shows the formation of an allopolyploid, and you can see that there are 2 different species, species A, which has 4 chromosomes, and species B, which has 6. And species A ends up producing an unreduced gamete because of a meiotic error, while species B produces 2 normal gametes. And this unreduced gamete will then fuse with the reduced gamete to form a hybrid that has 7 chromosomes. And this hybrid, because it has an uneven number of chromosomes, is not fertile. So in order to be fertile, it then goes through the allopolyploid fusion again. And this hybrid then produces an unreduced gamete with 7 chromosomes, and then this unreduced gamete ends up fusing with the second reduced gamete, original gamete from species B, in order to form the viable fertile allopolyploid hybrid. The second second type of polyploid is autopolyploid, and autopolyploids contain multiple chromosome sets derived from a single species, and they can also be formed by chromosome doubling or fusion, like allopolyploids. And this mechanism shows that there is a single parent species which then produces two unreduced gametes because of the meiotic error, and these unreduced gametes will then fuse together to form the autopolyploid zygote. Plants they found that there is polyploid evident in 50 to 70% of all flowering plants, and the majority of these plants, about 75% of the plants, are allopolyploids. And many agricultural plants such as cotton, oilseed rape, and wheat are allopolyploids. And some plants form triploid organisms, and because through the fusion of a reduced and unreduced gamete. And so in order to become fertile, it goes through the same process that I just showed you earlier for the allopolyploid. The uneven numbered chromosome hybrid will then produce an unreduced gamete and fuse with a reduced gamete in order to become fertile. Animals were thought not to undergo polyploidy because of their mode of sexual reproduction, but they have found that sex-determining mechanisms cannot explain polyploidization, and this is because there is an increase in cell size, and it can in turn alter the physiological or developmental processes that rely on a carefully balanced regulatory system. And also, animal cells will tend to lower their number of cells in order to cope with the larger cells that polyploid causes. And so, and this makes the polyploidization in animals more complex than it does in plants. And the formation of a polyploid in an animal is equal to the rate that unreduced gametes are produced and the pattern of mating that unite the gametes. And this also contributes to the complexity. There are a number of different mechanisms by which polyploids can be formed. Polyploids can be formed through sudden variations in physiological and environmental factors such as temperature change, and the temperature change causes chromosome doubling. And so L.F. Randolph did an experiment with corn, and he exposed the corn to a high temperature, about 40 degrees Celsius, for 24 hours. And after the exposure, he found that 1.8% of the corn was tetraploid, and 0.8% of the corn became octoploid. And another way through which polyploids can be formed is through gametic non-reduction. And this is when gametic non-reduction is when unreduced gametes are formed, and unreduced gametes contain the full somatic chromosome number, and then these gametes can either fuse with unreduced gametes or reduced gametes in order to form a polyploid embryo. And an obligate cell is just a cell that is classified as an obligate polyploid cell, which is during the cell's normal life, it will develop nuclear DNA content. And an example of this is a megakaryocyte, which I will discuss later on. They have found also that cells that become old and have great functional stress can cause polyploidy in the cells. And also, an uncommon way through which polyploidy can occur is through polyspermy. And this is when an egg is fertilized by 2 or more sperm. They have found that about 2 to 3% of human pregnancies, this occurs in about 2 to 3% of human pregnancies, and it results for about 15% of miscarriages. And if the baby ends up surviving to birth, it will die at an early age. Vivian Negron Ortiz did an experiment with the Cactaceae family, and she looked at this family because polyploidy is one of the major causes of speciation in Cactaceae, and she specifically looked at the Consuela species within this Cactaceae family, and she found that the most common polyploid form of the Consuela species is a hexaploid, so it has a 6N instead of the regular diploid 2N. And so she found that it has a common pathway, that the hexaploidy is formed with a common pathway that alfalfa, which is just a flowering plant and the same pathway as Beta vulgaris, which is like a bee. And they both form their hexaploidy through the one-step mechanism, so the fusion between unreduced and reduced gametes. And she also found that there is another way present in which the polyploidy of the Consuela species occurred, and this is through cytomixis. And cytomixis is a process of chromosome transfer that can occur during pollen formation, and it results in pollen grains that have chromosome number greater or less than the haploid number. Negron-Ortiz also found that endopolyploidy was present in the Consuela species, and this endopolyploidy is when chromosomes replicate without the division of the cell nucleus, which then causes a polyploid nucleus. And this is an extremely regulated process and occurs in active development. And she found that the endopolyploidy occurs in water storage cells during when there's a lot of rainfall in the desert. So the endopolyploidy will cause the water storage cells to increase in size. So when there's a lot of rainfall, this way the cells will be larger and the cactus can then take in more water and store more water in order to get through the harsh desert conditions. There are three mechanisms by which animals can become polyploidy, and this is through cell fusion, endoreplication, or the defects of the abortive cell cycle. Cell fusion is when two cells fuse together to form a polyploid organism. In this diagram, you can see that there are two diploid cells that end up fusing together in order to form a tetraploid cell. Endoreplication is the replication of the nuclear genome in the absence of cell division, which leads to an elevated number of gene content and polyploidy. And the cells become autopolyploid because they fuse together and continue to replicate its chromosomes. And in this diagram shows the normal cell division cycle, where there's the G1 phase where the cell grows, S phase where the chromosomes replicate, G2 phase, and then mitosis where the cells split. And with endoreplication, there are two subunits where there's endomitosis or endocycling. And in endomitosis, the cell will go through the G1 phase and then replicate its chromosomes in the S phase, and then go through the G2 phase. But then it will only go through part of mitosis, which then causes the cell to go back into either the G1 or S phase. And because it only goes through part of mitosis, the cell doesn't divide all the way, causing the cell to become polyploidy. And in endocycling, it just skips mitosis altogether, so the cell will go through a growth phase and then replicate its chromosomes, and then go straight back into the growth phase and then replicate. And so this is how this cell becomes polyploidy. And it is known that Drosophila embryo cells go through endocycling, so they skip mitosis altogether, and that is how they become polyploidy, where megakaryocytes go through endomitosis, so they skip some of the steps of mitosis. And there is not a specific or a certain part of mitosis that each of these cells will skip. Like, different cells could go in and just start in prophase and just do a little bit of prophase and then go straight back into the regular cell division cycle. Or the cell can go all the way through mitosis and just not finish cytokinesis and then go in. So there's not a set place where each of these cells stops. Megakaryocytes, as I stated before, are classified as obligate polyploid cells, and this is because they become polyploidy during their normal development. And the megakaryocyte switches from a regular mitotic cell division cycle to the endomitosis cell division cycle. And this makes the megakaryocyte become massive and polyploidy. And there is a block in prophase when the cell enters into mitosis, and the nuclear membrane doesn't disappear all the way, which causes the cell to become polyploidy and proceed back to the G1 state. They have found many different polyploid chromosome numbers and DNA content in the megakaryocyte. They found that a megakaryocyte can have 18N, 16N, 32N, 64N, and even 128N. But they found that the median DNA content of the megakaryocyte is 16N. There are many speculations as to why the megakaryocyte switches to endomitosis, and sources suggest that the reason is due to the reduction in activity of the cyclin B-dependent CDC2 kinase. And recent studies have found that acceleration or premature destruction of the cyclin B1 by the ubiquitin proteasome pathway allows the megakaryocyte to re-enter the S phase of the cell cycle. And J.M. Zimmett, he did a study on transgenic mice to look at this, and he found that there was an overexpression of cyclin D3, which this study then shows that cells are able to obtain a high level of ploidy because of the multiple cycles of endomitosis and DNA synthesis that the cell can go through. The last way in which animal cells can undergo polyploidy is if there are defects of the abortive cell cycle. And basically, the cell slips past its arrest. And this diagram shows 4 different ways in which a tetraploid cell can go through. There's a tetraploidy checkpoint, and in A, the tetraploid checkpoint works so it recognizes the tetraploidy, which then causes the cell to go back to the G1 phase and arrest. And in B, the checkpoint is compromised, and so that then the cell will proceed through division, but this most likely leads to aneuploidy because of the way that the chromosomes line up in the metaphysical plate. And so the aneuploidy will then lead to cell death. In C, this is known as reduction mitosis, and the diploid will then segregate to one pole, and then the other 2 will just go to opposite poles. And so basically the 4N then reduces down to the regular diploid 2N, and then these 2 become haploid and then end up going through apoptosis and die. In D, this is how many cancer cell types, this is the pathway through which many cancer cell types occur. And there are extra centrosomes that are clustered in the middle here, which allows the stable propagation of this cell as a tetraploid. Whereas, see, they are clustered here where they're not clustered on the outside, which the clustering causes the cell to be able to slip past the arrest and continue as a tetraploid. In conclusion, I think that it would be very beneficial to study this more in order to understand the total mechanisms, because most of them are not known. We just know that they undergo polyploidy. So the cells undergo polyploidy, but we're not really sure exactly why they do. And I also think that it would be beneficial to study animals in order to find out if there are other cells that undergo polyploidy and why they would need to undergo polyploidy. And there's a growing need for plant materials for food, feed, and fuels. And I think it would be beneficial for us to be able to manipulate the process of polyploidy in order to, which will help us to manipulate the gene expression and heterosis in the hybrid plants, and as the need for the food, feed, and fuels grow. Any questions?